Okay, in the meantime, good morning, everybody. I'm Fransiska Malfait. I'm a clinical geneticist, and I work in the Ghent University Hospital in Belgium. And so, yes, I'm between the interesting topic, on sex and drugs, but in the meantime, I have to talk a little bit about DNA. I'm also the CSO of the EDS Society. Let's see if that works, yes. Okay, so DNA. Of course, our DNA is the molecule that contains our hereditary material, and thus also the biological instructions that make all of us very unique. So, of course, our bodies are very, very complex structures, and so the DNA contains the code for making this body. However, when we look at our DNA in detail, it's very surprisingly simple. So, we all know that this DNA is this double-stranded helix, but when you look in detail, you see that it only contains 4 different building blocks, the letters of our DNA, and so these are A, T, G, and C. And so every molecule that is made in our body is based on this 4-letter code. So just a fun fact about DNA. So every cell in our body contains the full hereditary material. So it contains the full length of our DNA. And so that's about 2 meters in every cell. And if you know that we have so many cells as shown there, 10 to the 14th, I don't even know how many cells that are. There are a lot of zeros in there. But then you multiply these 2 meters by the number of cells, then you know that you have enough DNA in your body to stretch back and forth between the Earth and the Sun 1,000 times. I think that's just amazing, really. So if, we know that our DNA is this long, we also know that there is a lot of opportunity for variation in our DNA. So our body really has come up with a very clever way of packing this very long structure in all these tiny cells. And so the DNA is packed in these structures called chromosomes that you see there. And so each cell in our body contains 46 chromosomes. And so that's where our DNA, this double helix, is packed. So the genes what are genes actually? Genes are just parts on our DNA, stretches on our DNA that contain the instructions for making proteins. And the proteins are actually the building blocks of our bodies. For instance, collagens is a very important protein in our connective tissue. So that's a protein. And so for each type of collagen, for instance, there is a specific stretch on our DNA that codes for the making of that collagen. And that's called then a gene, a collagen gene. So you could actually say that our DNA is a cookbook, whereas the gene is a recipe for a specific protein. So the DNA is the complete cookbook, the gene is the specific recipe, and then the protein could be, for instance, an apple pie, but in real life, for instance, a collagen. So when we look at our DNA, actually, so only about 2% of that full length of DNA that we find in every cell codes for the making of proteins. And then the other 98%, we actually don't know yet what that DNA does, or we don't have a full view on what that does. And for a long time, this other part of our DNA was called junk DNA. But the one that we actually mostly focus on in clinical genetics is only that 2% that codes for the making of proteins. And why? That is because most of the defects that we find now leading to hereditary diseases, such as, for instance, certain types of Ehlers-Danlos syndromes, are found somewhere in that 2% of the genome that codes for the making of proteins. So the factories in our bodies that actually make those proteins based on the code in the genes are the cells. And we have many different types of cells in our body. And you can see they also are different in shape depending on the organ they function in. For instance, we have the blood cells, we have bone cells, we have sperm cells, we have motor neurons. And so, like I said, every cell, all these different types of cells, they contain the complete copy of our genome, of our genetic material. But in contrast, not every gene is actually translated or made into a protein by each cell. Each type of cell will only make those proteins that it needs to ensure its function. So in our connective tissue, let's see, yeah, so like you say, one cell would make a fruit cake where the other cell would make a fish dish, if we continue along the recipe metaphor. So the Ehlers-Danlos syndrome, what we know of it is that it's a connective tissue disease. So it's the cells of our connective tissue that are not functioning well. And so the main cell in our connective tissue is called a fibroblast. So these are the cells that we find, for instance, in our skin, but also in tendons and ligaments, for instance. And so these fibroblasts, what they mainly make are these types of proteins that you see there. So collagens, but then there are also a few other ones, such as proteoglycans, glycoproteins, elastin, etc. And all these proteins are the main building blocks of our connective tissues that we find in bone, cartilage, tendon, the blood vessel wall, for instance, but also the wall of our gastrointestinal tract, the skin, and so on. And so when you have a defect in a gene that codes for one of these proteins, such as a collagen, it's no surprise that you will have abnormalities or defective function of all these different tissues, such as tendon, skin, blood vessel wall, etc. Now, we're talking about genetic variation here and variants of unknown significance. But what is genetic variation? Genetic variation is a term that is used to describe the variation in a DNA sequence that is present in each of our genomes. It's what makes us unique, whether in terms of skin color, hair color, eye color, the shape of our faces, our stature, everything that determines on. So we all have variations in our DNA that makes us different from other people. And so not all variation, of course, is disease causing. Most of the variation is not disease causing. So what does that variation mean when we really look at the molecular level in our DNA? It means that, remember I talked about this 4-letter code, it means that at a certain position in the DNA, one of those letters, you can see there on top you see CG, where on the lower part you see TA, if you can see it, I don't have a pointer, but it means that at a certain position, the letter in my DNA will be different than the letter in your DNA. And that might determine that I have a different color of my eyes or a different color of my hair, for instance. When this is not disease-causing, we call this a single nucleotide polymorphism, or just in short, a SNP. Okay? And so here, when there is a SNP in your DNA, it's depicted by the different colors. So the different colors just mean that there is a slight variation in the same copy of a certain gene. That is not disease-causing, it just makes it a little bit different, okay? So SNPs occur normally throughout a person's DNA, and so they occur almost once in every 1,000 DNA letter, which means that there are roughly about 4 to 5 million SNPs in a person's genome. So when we do genetic analysis of a bunch of genes or of really the whole genome, we will pick all these variations up. And sometimes it's quite difficult to determine when we find a variation whether it's just a normal SNP or whether it's really disease-causing. And so these variations may be unique or they may also occur in many individuals. And so the more genetic analysis we do, the more of these variations are uncovered in populations around the world. So when we do genetic analysis in a person, sometimes we do find a new variant of a gene with a new SNP, and then sometimes it's quite difficult to interpret. Sometimes it's not difficult to interpret. Sometimes we really know, okay, the variation at this certain position is not going to interfere with the function of the protein that is encoded by this gene. We know that the protein will have a normal function, a normal quantity, so not disease-causing, and then we call this a benign variant. Sometimes we are absolutely sure when we find a variation in a gene that it will lead to an abnormal protein or, for instance, also to an absent protein. And then we know that we have a problem. And this is something that will be disease-causing because we know that the protein function is not ensured. And so then we call this a pathogenic variant or a mutation. A mutation is a change in our DNA sequence that changes thus the function of a protein. It can be either due to a mistake when the DNA is copied, and this is something that, for instance, you will see in hereditary diseases that we inherit from our parents, in some cases of EDS, for instance. Could also be the result of environmental factors such as UV light or cigarette smoke. This is something that you would see in diseases such as cancer. Okay. However, sometimes we don't know whether a new variant that we pick up by genetic analysis will either lead to a normal protein or to an abnormal protein or an absent protein. So we don't know, and that's when we talk about a variant of unknown significance or a variant of uncertain significance. It's a VUS, so this means it's a variant form of a gene, a new form of a gene that we identify through genetic testing, but whose significance to the function or the health of an organism is not known. So we simply don't know. And of course, when you find a variant in a gene, you know, of interest for your disease, it's very tempting to think that it will actually mean something, that it will be disease-causing, but we have to be very careful with this interpretation because sometimes it's not, and that over-interpreting or misinterpreting this variant of unknown significance can also have very harmful consequences. So in genetics, when we do genetic analysis and we find variants in genes, we use this classification for variants. So we say it's benign or likely benign, and then it means that it means nothing. It's not disease-causing. So benign and likely benign variants will not be reported in your genetic test. On the other side of the spectrum, you have pathogenic or likely pathogenic variants, and there we are sure that they will be disease-causing, and so they will be reported as being the cause of a hereditary disease. And then you have the uncertain, the VUSs, uncertain significance. Usually they are also reported in your genetic test. But again, it does not mean that this is the cause of your disease. We just simply do not know. And then sometimes, but not always, you can do additional studies to try to reclassify this variant either to benign or likely benign, or to pathogenic or likely pathogenic. And what are these additional studies you can do? Well, here you can see a few. You can do family studies, and I will explain what I mean by this. We can do literature research. We can see in the medical literature whether the same variant, for instance, has been reported in other families and other patients, and whether those families had the same clinical characteristics, same disease as the patient that you're studying. We can also look in public databases, and I will also explain what I mean by this. And sometimes, but not always, we can do functional studies. For instance, for EDS, mostly those functional studies would be done on a skin biopsy, because on a skin biopsy, we can actually extract the proteins that are encoded by those genes. For instance, collagens, they are expressed in the skin, and then we can do collagen studies. And then for a very few types of EDS, there is also a very specific urine test, but that's only for a very rare type of EDS. So first of all, family studies. What can you do by examining families? Now, let's say that you have a person here, this is the lowest person, the circle with the arrowhead there. Let's say that this person is affected by, has signs of Ehlers-Danlos syndrome, and you do a genetic analysis of all the EDS genes, and you find a variant of unknown significance in one of the EDS genes, but we don't know what it means. You can see that in this pedigree, so you have the squares are men, the circles are women, so you have the parents and the grandparents, and then also an uncle there. The symbols that are colored mean that this person is also affected by the same clinical characteristics as the one with the arrowhead. So it means that her father also has signs of EDS, and her paternal grandmother also has signs of EDS, and the other ones do not. So what we then typically do is we try to get also a DNA sample of other people in the family. And let's say that you would start with the grandmother who is also affected, and you look at this presence or absence of this VUS in the same gene. You can see here that this grandmother has the same VUS. So this could still mean that this is a disease-causing variant, because there's 2 affected people who have that specific variant. However, if we would then take blood and a DNA sample from her father in between there and the unaffected uncle, the brother of the father, you see that the unaffected one also has this VUS, whereas the affected one does not have this VUS. Well, by doing this, we are now sure that this variant of unknown significance cannot be the explanation of the disease, because the affected father does not have this VUS. And so by doing this, this would enable us to reclassify this VUS as benign, so not disease-causing, because one of the affected individuals does not have this VUS. So in this way, family studies can help you in interpreting the meaning of a VUS, but of course you would have, you need then also a DNA sample and a very good clinical characterization of the people that you would investigate. Then I told you we can also look in public databases. And just as an example, as geneticists, we have a few databases that are freely available online and that we have access to, such as, for instance, this ClinVar. I'm not going into too much detail, but just to show you, for instance, ClinVar is a freely accessible public archive of reports of the relationship among human variation and the clinical phenotype with the clinical characteristic with supporting evidence. So when scientists do, or doctors do genetic analysis all over the world, they will put all that information into these public databases together with clinical information so that other scientists and doctors have access to this information. And by doing this, we gain more knowledge about the genetic variation and the clinical characteristics that are associated with this variation. And so we can look up when we find a new variant, has it been reported in other populations? And if so, what were the clinical consequences? And then we can compare that with our patient. Sometimes we can also do functional studies, like I said. We can usually we do that on a skin biopsy for Ehlers-Danlos syndrome, and then we can culture the fibroblasts and the main type of cells that we get from the connective tissue, and those, for instance, produce collagens, but also other EDS-associated proteins, such as, for instance, tenascin X. We can extract those proteins, and just as an example, for quite common type of protein analysis we do in EDS is a collagen biochemical test. There you can actually see the proteins of the collagens, and then this is a very clear example. You see 3 lanes in the 2 pictures, and in the middle lane you see that the lower band there is not present. It's actually the absence of a certain type of collagen, and that really shows you that no product is made by a certain gene. So this is just as an example of a functional test that you can do. However, it really depends on the gene in which the VUS is found, whether you can do functional studies and which type of study you can do. So that's really not one test for all type of VUSs. And in reality, a functional analysis is not available for every identified VUS. So when additional studies actually do not allow to reclassify a variant as benign or as pathogenic, it can remain a VUS actually for a long time. So sometimes we simply do not have a final answer. But as we test more and more people throughout the world, and by putting all that information also in these public databases, we learn more and more about the significance of these VUSs, and maybe in a few years' time, when we have more information, we will be able to reclassify them as pathogenic and others as benign. So therefore, when a VUS is identified, it is also important to stay connected with a geneticist or a counselor who can also keep you updated on these results. So yeah, that's it. I hope that I was able to clarify this a little bit, how we tackle this problem of VUSs, and I would be happy to discuss this further during the Q&A or over a beer, maybe later. Thank you.